Start recording. Share the screen. I don't think this is working, so I'm going to have to, oh, there it is. It did show up finally. All right, so we'll work a little bit on lab 8. We're about a half an hour, not lab 8, lesson 8. We're about a half an hour. And then we will do lab 9. Please note that your infectious disease paper is due 11:59 p.m. this Saturday. All right, any questions? All right, let's begin chapter 8. I'll keep that up in case I need it. So we had talked about the lac operon, and this is an inducible system that bacteria have, particularly E. coli, for controlling the expression of genes. Any question about the lac operon or gene control? Not hearing anything, so I'm going to move forward. Let's move into the topic of mutation. Mutation is a change in the genetic material. So when you have a gene and it's encoded by a certain sequence of nucleotides, if you change that sequence of nucleotides, that results in a mutation. Mutations may be neutral, beneficial, or harmful. Here we're looking at a mutant I, normal I. And I can mention that the mutation, I don't think I've talked about it in this class, but maybe I did. Everything runs together after a while, so I don't remember what I've talked about and whatnot. But 10,000 years ago, the human population had a mutation in Northern Europe, giving rise to the eye color blue. And back then, it appears that blue eyes had a selective advantage because blue eyes is now the major allele in Northern Europe. Today, when we look at eye color, blue versus brown, or any other shade of those two, we don't find any selection for one or the other. There's nothing beneficial about having blue eyes over brown eyes. But it's possible that 10,000 years ago, there was an advantage to having blue eyes. And that would explain why blue eyes became the predominant allele in Northern Europe. It's also possible that blue eyes was next to an advantage, a gene that gave an advantage, and then blue eyes was carried just because it was close to a gene that was being selected for. Anyone have any questions about that? The point is, over 10,000 years ago, we only had humans with one eye color, and that was brown-eyed humans. And there'd be different shades of brown, but everyone had brownish eyes. And then 10,000 years ago, there was a mutation giving rise to blue eyes. The question is, will there be a mutation in the future that will give humans another eye color? Because if you don't know, dogs have blue eyes, they've got brown eyes, and they've got yellow. And my guess is that sometime in the future, we probably will get yellow eyes. All right, any question about any of that? A mutagen is an agent that causes mutations. Spontaneous mutation is when the mutation occurs in the absence of a mutagen. Spontaneous mutation happens all the time. And what happens is the DNA polymerase makes a mistake and then that can result in a spontaneous mutation. It doesn't happen very often, meaning most of the time the DNA polymerase works fine, but every once in a while it can make a mistake. And when you're talking about the total number of nucleotides in a in a person, and then the total number of people on the planet Earth, you can see that spontaneous mutations are happening. Okay, any question about any of that? There are different types of mutations. There's a base substitution, also called the point mutation. And that's where one nucleotide base gets changed. You can have different types of base substitution. You can have a silent mutation, a missense mutation, or a nonsense mutation. You can also have a frame shift mutation. That is where a nucleotide gets added or subtracted from the sequence of nucleotides. And when you have a frame shift mutation, you change everything downstream of the frame shift mutation. Because as we mentioned, messenger RNA is coded in codons. And if you pull out one nucleotide base, then the next codon, instead of being read three, there will only be 2 there. And so it'll borrow the next nucleotide. And so all the codon is changed after the frame shift mutation. Is there confusion about a frame shift mutation? Have I explained that correctly? Or good enough? All right, and there's also a macromutation. Pay attention to my lesson and my notes on a macromutation, because a macromutation is not discussed in the textbook. A macromutation is where an entire region of a chromosome is either added or deleted. And that results in usually a large number of genes being changed. Does anyone know a macromutation where the entire chromosome is extra in a person?
Down syndrome.
Yeah, Down syndrome has 3 chromosomes, 23, I think it is, 21. 21? Nobody's correcting me, so I'm going to assume it's 21. That would be a macromutation, where every gene on that chromosome is going to have an extra copy in the cells. And normally, when a human has an extra chromosome or a missing chromosome, it's lethal. But in the case of Down syndrome, it's a very small chromosome, and it changes the individual, giving them a Down's phenotype, but it is not lethal. There are a few other examples of that, usually with the X or the Y chromosome, meaning there are people born with only 1 X chromosome, and there are people born with 3 X chromosomes, and those are macromutations. There are regions of the chromosome that can be added or deleted, and those are not usually, well, it depends on the region, I guess. It could be lethal, but sometimes the patient can survive. Here's a case, I've never read anything further about it, where scientists were checking this boy. I don't know if you can see it. They were wondering if he was a mutant, because as you can see, metal spoons were attaching to his skin. And people were wondering, is he a magneto? Anyways, I only read that they were looking into whether he was a mutant or not. I never read anything further about it. Mutation can occur as a base substitution, where when the DNA is being made, the DNA polymerase makes a mistake. So it puts in A because there's a T over here, but instead of putting the C here, it puts a T. That, if it's not corrected, will give rise to a mutation, because when this strand is now replicated, instead of being a GC, which is normal, this strand will become an AT. And that codes for a different codon. That's not terribly important at this point. But the point is that would give rise to a mutation if it's not corrected. Any question about any of that?
Does it cause a specific type of mutation when that happens, or just an overall mutation?
Well, it's a single base that's mutated, so it's a point mutation. But there's different types of point mutations. We'll discuss that in a minute.
Okay, thank you.
Okay, all right. Is that good enough? Good. The first mutation I want to talk about is a silent mutation, sometimes called a neutral mutation. It is a base substitution which changes the DNA. One base in the DNA is changed. However, when it's transcribed into RNA and then translated into protein, the protein is not changed because you're changing the nucleotide, but you can change it to another codon that codes for that same amino acid. For example, if UUU, which is the RNA, were to be changed to UUC, it still codes for the amino acid phenylalanine. And the DNA would be AAA or AAG. And although that's a change in the DNA, it does not result in a change in the protein. So there's no effect on the protein. So this they call a silent mutation. Any question about any of that? All right. There's also a missense mutation, and that is where you change one base in the DNA and it results in a change in one amino acid. So AAA codes for UUU is the codon, which codes for phenylalanine, the amino acid. And if we were to change that, it looks like I've got that wrong. I should be going CCG, which codes for glycine. And then we change that from CCG to TCG. There's only one nucleotide change. The C is changed to a T there. And that causes a change in one amino acid in the protein. This is called a missense mutation, where you change one nucleotide base and you change one amino acid in the protein. Any question about that? An example of a missense mutation is sickle cell anemia, where the patient usually only has one amino acid differing out of thousands of amino acids in hemoglobin. And that gives rise to the sickle cell anemic trait, assuming the person has two of those genes. Another example of a missense mutation was in Northern Europe 10,000 years ago. We had a single base change, and it gave rise to blue eyes in the human population. Okay. Sickle cell anemia is a harmful gene, so the person born with it is deleterious effects, assuming they're born with both of those genes. The blue-eye gene, as far as we know today, is a neutral mutation. Any questions? They're both missense mutations, though, because they change the DNA by one nucleotide, and they change the protein by one amino acid. A nonsense mutation, let me get that out of the way, is where you change one nucleotide base in the DNA and you change the codon. Instead of coding for an amino acid, it codes for the stop codon. And this is called the nonsense mutation. What happens with a nonsense mutation is you'll get a truncated protein. Instead of a protein, this one only has 4 amino acids, which I don't think there's any protein with that short. And this one only has a protein with 1 amino acid. So after the stop codon, nothing else is translated into amino acids. And so after the stop, you get no further protein. The protein is truncated. Any questions about a nonsense mutation? All 3 of these, the silent mutation, the missense, and the nonsense, is a point mutation where you're only changing one nucleotide base. A frame shift mutation is where you insert an extra base. I'm sorry, what's the question? Somebody might have their microphone on, so if you did that unintentionally, please turn your microphone off. A frame shift mutation is where you add one nucleotide base or you remove one nucleotide base. So normally we have the amino acids AAG for lysine, UUU for phenylalanine, GGC for glycine. And here we're pulling out one amino acid. The A is being pulled out, so we'll, instead of getting a U down here, the codon then reads the next G. So we have the AAA, that's before the frame shift mutation, or is it the AAG, before the frame shift mutation? So that's the same. And then we have UU, and then it reads the next nucleotide, which will be a G, and that's going to change this amino acid. Instead of phenylalanine, it will then code for leucine. But because we remove one nucleotide, the codon is not going to be GGC anymore. It's going to be GCU. So every point after this frame shift, the codons are going to change. Any question about that? Let me see if I can shut somebody's microphone off. There, I got it. All right. So after a frame shift mutation, everything downstream of the frame shift will be changed. And that's the case whenever you have an insertion or a deletion. There is an exception though. If you have an insertion of 3 nucleotides or a deletion of 3 nucleotides, you only change one amino acid. For example, if we were to remove UUU, you would, instead of going methionine, lysine, phenylalanine, glycine, you would go methionine, lysine, and then to glycine. Any questions about frame shift mutation? The frame shift mutation, as well as the I'm trying to show it. The nonsense mutation really really make the protein different. Oftentimes after the nonsense mutation or after the frame shift mutation, the protein will be in the wrong configuration, and so it won't work. Some examples of frame shift mutation are some Tay-Sachs patients have a frame shift mutation. All right, any questions about mutations? That's all of them, except for, as I mentioned, the macromutation is a region of the chromosome which is either added or subtracted. And it can be the entire chromosome in the case of Down syndrome or individuals who are hermaphrodites, XY individuals. Any questions about mutations? All right. All right, so mutations can be spontaneous or they can be caused by mutagens. Spontaneous mutation, as I stated, happens all the time when the DNA is simply copying the nucleotides. Every once in a while, it's not very often, something like once in a billion times, or maybe even less than that, it makes a mistake. And a mutagen is where something increases the mutation rate, where a chemical or some other agent causes the mutation. There are two types of mutagens. There's radiation and there's chemical mutagens. Ionizing radiation like X-rays and gamma rays has high energy, and that causes the formation of ions. And then that ion can react with DNA, causing a mutation. So X-rays and gamma rays, when they hit an atom or a molecule, they will hit the electron and then give the electron so much energy that the electron will escape from that atom or molecule. So it'll go elsewhere. And then this will be an ion here. You'll have a positive charge. And then this molecule can react with DNA and cause a mutation. So that's how ionizing radiation can cause a mutation. Any question about that? There's also UV radiation, and it causes a mutation in a different way. Let me see if I can blow this up. The UV light, like from the sun, can hit your skin. And whenever there's two thymines adjacent to each other, when UV hits them, those thymines that are adjacent to each other can form a covalent bond between them. And it's actually two covalent bonds between them. So we call it a thymine dimer. When a thymine dimer happens, this causes a bubble in the DNA. The two thymines do not hydrogen bond with their corresponding A's over here on the other strand. And then they bond to each other, and that doesn't normally happen where DNA has a covalent bond between them. And wherever this bubble is, the DNA cannot be replicated or transcribed. And so if the DNA wants to be replicated or transcribed, the cell has to repair this. You get too many of these in the cell, it'll be lethal to the cell. Any question about UV radiation and how it causes problems for DNA? All right, if not, let's talk about chemical mutagens. We're only going to talk about it with nitrous oxide, nitrous acid, which is a mutagen. When nitrous acid combines with DNA, it changes the DNA. This A here goes CGA. This A gets chemically modified by nitrous oxide. And it is similar to an A, but we're going to call it a modified A, meaning it's red. And when the DNA gets replicated, the DNA will read this strand fine because it's not mutated. But this one here has been mutated by the nitrous acid. And that A is modified, and the DNA polymerase will not read it as an A. It will read it as a G. And so, as opposed to this modified A, the DNA polymerase will put a C there. And then this will result in a mutation when this gets replicated, because this will now encode in, instead of coding for AT, it'll code for GC. And that A is going to be acting like a G. Okay, any questions about how nitrous acid can act as a mutagen? All right. If there's no questions, I think we'll stop the lesson here. We've now talked about mutation. We'll have to talk about the frequency of it, but that we can do next time. Oops, too far. All right, if there's no questions, we'll begin with the lab. We're going to be discussing lab 9, the identification of gram-negative organisms. Come on. I want that actually, but I want to shut that down. Oh, I did shut it down. Never mind. It just took its time doing it. All right, lab 9. Let me make sure I got the right one here. Yep, that's the right one. Identification of gram-negative bacilli. There is some reading you should do beforehand. Not very much this time, you'll be pleased to know. Upon successful completion of this lab, students should be able to interpret the results of the following biochemical tests. The IMVIC test, which IMVIC is an acronym for indole, methyl red, Voges perspire, and citrate. You simply take the first letter of each of these, and then add the I before the C to get IMVIC. So understand these tests and be able to interpret them. You should also know about carbohydrate fermentation, which we discussed in the lab. We did PR lactose before. You should know about the motility test and hydrogen sulfide production test. Be able to explain how to interpret triple sugar iron slant, which is also called the TSI test, TSI for the acronym triple sugar iron. The SEM deep, and the SEM is an acronym for sulfur indole motility test. And then growth on McConkey's algae, and heptone enteric algae. And then lastly, be able to use the biochemical analysis to solve case studies involving gram-negative bacterial infections. Members of the family Enterobacteriaceae are small gram-negative, non-spore-forming bacilli that can be found almost everywhere. They are facultative anaerobes. There are 53 genera in this family. 24 of these genera are known to cause human infections. All of them are glucose fermenting oxidase negative organisms. And here you're seeing some of the families of the some of the genera of the family Enterobacteriaceae. Most of them are normal microbiota of the intestinal tract. So we call them coliforms, like, where is it? Escherichia, we found in our intestines. Salmonella is not in our intestines, but it is in the intestines of birds and probably turtles, things like that. They're opportunistic pathogens in that they can cause urinary tract infections, wound infections, pneumonia, septicemia, that's an infection of the blood. Common genera responsible for causing opportunistic infections include Escherichia, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several of these genera are associated with gastroenteritis, and here they are frank human pathogens. These include the genera Salmonella, Shigella, and certain variants of E. coli, like E. coli serovar 0157H7. Any question about any of that? Another of the gram-negatives we have to talk about are the Pseudomonas, Pseudomonaceae family. There's one member in particular, Pseudomonas aeruginosa. They're similar to the family Enterobacteriaceae in that they're gram-negative bacilli. However, all of the Pseudomonas do not ferment glucose. So we call them non-glucose fermenting. They're oxidase positive. And those are the two ways they differ between Enterobacteriaceae and Pseudomonaceae. Pseudomonas aeruginosa can cause opportunistic pathogens, particularly in the immunosuppressed individuals, burn patients, and trauma patients. Pseudomonas aeruginosa is also an infection of health care acquired infections, also called the nosocomial infection. It's important for a microbiologist to be able to identify and perform antibiotic susceptibility testing on gram-negative bacilli cultured from a clinical specimen. In this lab, common biochemical tests will be discussed that are used to identify gram-negative bacilli. These are the biochemical tests you need to know. The indole test, some organisms produce the enzyme tryptophanase, which convert the amino acid tryptophan into the products indole and pyruvic acid. After overnight incubation, Kovacs reagent is added, which reacts with indole to produce a red color at the surface. E. coli tests positive for indole. Klebsiella pneumoniae does not. So here we have tryptophan in the presence of tryptophanase. Indole gets made, and that doesn't look very red. That's just the photograph, but it's going to be at the top a reddish, or in this case, kind of a purplish color. And then the negative will not be reddish. It'll be yellowish. All right, any questions about the indole test? Remember with the indole test, you take the solution that the bacteria is grown in, and then you add Kovacs reagent. Kovacs reagent will float on top of the aqueous media. And that's why that's going to be on top. Methyl red and Voges Prescure are two tests, but they start from 1, 2. So to run the MR and the VP test, the Voges Prescure, you inoculate 1 tube, which will be MR VP tube, and then you grow the bacteria, and then you divide that tube into 2 portions. One portion will be used for the methyl red test, also called the MR test. The other test will be, the other portion of the tube will be running the Voges Prescure test or the VP test. So you start as 1 tube, and then it'll divide into 2 tests. Any question about any of that? The MR test and the Voges Prescure test is looking at the results of glycolysis, such as fermentation and glycolysis. And if the glycolysis produces acid products, the MR test can distinguish it. So the MR test will be looking for acid end products in glycolysis. The VP test will be looking at neutral products from the glycolysis of glucose. The MR VP test is incubated for 2 to 5 days. MR requires 5 days. If you run it for 2 days and it's negative, you then have to run it for 5 days. And so if you ask for that test in the unknown, I will not give it to you until 5 days. Methyl red, you take the MRVP test and you add your bacteria to it. And after 5 days incubation, you then take the tube and divide it into 2. And you add methyl red to the MR portion of the tube. And if it turns red, the MR test is positive. If it does not turn red, stays yellow, then the MR test is negative. Any question about that? The VP test is used on the other half of the tube. And what you do is you add Barrett's reagent A and Barrett's reagent B to it, shake the tube to add oxygen, and then let it sit for 30 to 60 minutes. And if acetone was produced in the tube, the reagents will act with it and turn red. So in the VP test, the Voges Prescure test, once again, if it turns red, it's VP positive. And if it does not turn red, stays yellow, it's VP negative. So in all cases, indole, MR, and VP, if it's positive, the tube will turn red. Any question about that? And if it's negative, the tube will stay yellow or turn yellow. All right, the next part of the IMVIC test is the citric utilization test. This test is used to determine whether bacteria have the ability to use citrate. Citrate is a sugar that is found in the Krebs cycle. And if citrate is the only carbon source in the tube, if the bacteria can use it, they will grow in the tube. And then the tube will turn blue from the pH indicator bromo-thymo blue. The tube turns blue when the tube becomes basic. So if the bacteria can utilize citrate, it will turn the tube blue. That's from the bromothymol blue, which becomes blue color when the tube becomes basic. If the bacteria cannot grow on citrate, then there'll be no growth in the tube, and the tube will keep its green color. Okay? So I've got up there the E. coli and Shigella metabolize citrate, and they'll turn the tube blue. I don't have an example. Well, Pseudomonas will not grow on the tube, and it will be green. All right, carbohydrate metabolism test is looking at the fermentation of the sugar, and therefore we call it the carbohydrate fermentation test or the carbohydrate metabolism test. Each tube has one sugar in it, and it could be glucose, lactose, sucrose, maltose, etc. And if it can ferment that sugar, acid products will be made, and the acid will cause the phenol red pH indicator to turn yellow. And so if the sugar is fermented, it'll turn yellow, and that may be without gas being produced in the fermentation, or it could be with gas produced in the fermentation. Let me move this over here. So the tube has in it an inverted Durham tube, which you're seeing right there. And if gas is produced in fermentation, you'll see a bubble forming in the tube. This we call acid and gas, or AG for this result. This result is A for acid. This result is negative, no fermentation of the sugar. Each of these carbohydrate metabolism tests is a separate test, meaning if you do PR glucose and PR lactose, that is 2 tests for your unknown project. All right, any question about the carbohydrate metabolism test? We can mention that all members of the family Enterobacteriaceae will ferment glucose and turn the tubes yellow. Some members can ferment it without the production of gas, and other members will ferment the sugar with the production of gas, like E. coli will ferment glucose and release gas. Pseudomonas aeruginosa cannot ferment the sugar glucose, and so it'll be negative in this test. Any questions about carbohydrate metabolism test? All right, if not, let's talk about the motility test. Some bacteria possess a flagella, and that allows them to be motile and swim. Other bacteria lack the flagella, and they are non-motile. In a solid agar media, the agar is too thick for the bacteria to grow. But in the motility test, we have the agar at a very low concentration, and it's semi-fluid, and the bacteria can swim through the semi-solid agar media. In the motility test, if the bacteria are non-motile, you stab inoculate, and the bacteria are only seen growing in the stab. So this bacteria is non-motile. If on the other hand, the bacteria is motile, you'll see the bacteria flaring out from the stab. Or if the bacteria is very motile, the bacteria will go everywhere in the tube. Let me see if I can blow this up. And if it goes everywhere in the tube, the bacteria will cause the tube to look cloudy. If you notice here, this looks kind of clear and dark in this region of the tube. And here, the tube looks cloudy, and that's from the bacteria going everywhere in the tube. So this bacteria is motile. E. coli is usually motile bacteria. And let me see, I'm thinking Streptococcus pyogenes, but I really want to gram-negative bacteria. I'm not off at the top of my head remembering any Enterobacteriaceae, which is non-motile. I know they exist, but I don't remember one off the top of my head. All right, any question about the motility test? There are two tests for looking for motility. You could do the motility auger test, or the motility medium test, which all it is regular media with a low dose of auger. You could also do the motility test in the SIMS test. And we'll talk about the SIMS test in just a minute. The hydrogen sulfide production test is where you're looking to see if the bacteria can produce hydrogen sulfide gas from sulfur in the media. And the hydrogen sulfide test can be done on either the triple sugar iron, which is also TSI, auger slant, or the sulfide indole motility, and I call it the SIMS deep test, or the hectone enteric auger plate. All three of these media can be used to detect the presence of hydrogen sulfide gas. And all three of them turn the same color. And that is, if hydrogen sulfate is produced, the media will turn black from a precipitate occurring in the tube. And Citrobacter and Salmonella and Proteus produce hydrogen sulfide gas, and they will cause the tube to turn black. This is a SIMS tube, and you'll note that only the bottom is black. Hydrogen sulfide is only produced in anaerobic situations. So on the top of the tube, which is yellow, there's oxygen there, and you'll get no hydrogen sulfide production at the top of the tube. Normally, it's not this much. It's usually half the tube or the lower portion of the tube. But in this case, it's most of the tube. And there's hydrogen sulfide positive, hydrogen sulfide negative. This can also be tested on the triple sugar iron tube, and I'll show you that in a minute, or on the hectone enteric auger. Now here we go, hectone or enteric auger. You'll note that on this side, we have black colonies. That's because they're producing hydrogen sulfide gas. On this side of the HE plate, we have non-black colonies, they're white, and that's because they're not producing hydrogen sulfide gas. Any question about any of that? All right, if not, let's talk about the triple sugar iron test, also called TSI. This is a slant that you both stab, inoculate, and that should go down 2/3 of the way. That looks like it's only gone about halfway from this portion, the lowest part of the slant and the tube. This we call the butt of the TSI tube, and this portion the slant. And you inoculate, stab inoculate with the needle, the butt portion of the TSI slant, and then you take the slant out and then get more bacteria on it, and you put bacteria on the slant. This allows the bacteria to grow under aerobic conditions when it's growing on the slant, and under anaerobic conditions when it is growing in the butt down here. TSI agar slant is a complex media that has 3 sugars in it. Glucose at only 0.1%, and then lactose and sucrose at 1%. There's a phenol red pH indicator and peptone in it, as well as sodium thiosulfate, which is what turns the hydrogen sulfide black. All right, so you inoculate the triple sugar iron tubes, and then you look at the table for interpreting it. Table 1 has no fermentation of the sugar on the slant, so it's negative here, and no fermentation in the butt, so we say K, or you could say negative, negative. The first being for the slant, the second being from the butt. This one is complicated to discuss. Let's discuss it later. Let's move on to this one. This tube has fermentation of the sugar in both the slant, it's yellow, and in the butt. And so on tube 4, we'd say that it's AA, fermentation on the slant, fermentation on the butt, producing yellow being acid was AA. We can say that glucose is fermented for sure, or metabolized for sure, but one other sugar is also, and that's lactose or sucrose. We don't know which, and it could be both of the sugars. And the reason is because it's yellow in both the slant and the butt, and so we know one other sugar is fermented. In this case, gas is produced. That's why the agar is elevated and the agar is broken there. Let's see if I can blow that up so you see it's broken. See the cracking of the agar? That's because gas was produced. It's not black, so no hydrogen sulfide was produced. Let's talk about this one here. Oops. This one here is black in the bottom of the tube, not in the slant. So we can say hydrogen sulfide was produced. Hydrogen sulfide is only made when a sugar is fermented. So if hydrogen sulfide is there, you can assume the color of the media is yellow on the butt. You can't see it because black will obviously hide the yellow color, but it is there, so we can say that glucose is fermented. On the slant, it's red, and the reason why it's red was because all of the glucose was fermented here, but glucose is at lower concentration than the other sugars. It's only 0.1% glucose, and the glucose will get used up. And then the bacteria will start using the proteins for metabolism, and that will lower the pH. But initially, if we would looked at this within the first day, the slant would be yellow earlier than 24 hours. But all of the glucose gets used up, and then the protein is used, and that produces ammonia, which is basic, and that will change the color, changing it from yellow, changing it back to orange or pinkish color. And so this we say only glucose is fermented, and glucose will be yellow down here where it's black, but will get used up here because where the glucose is fermented anaerobically, it's going to ferment slower, and so the color will remain yellow. But on the slant, the glucose will be fermented very quickly. And so all the glucose will be used up, and it will change from yellow to orange and then pink as more basic products are made from the metabolism of protein. Okay, so that explains tube 2. Tube 3, we have glucose is fermented, why it's yellow in the butt, and then only glucose is fermented, why it's red or pinkish in the slant. The glucose got used up, the proteins were metabolized, producing a base, ammonia, which will change the pH, and it'll go from yellow to orange and then pinkish color. So that explains tube 1, tube 2, tube 3, and tube 4. Any questions about TSI? All right. You do not need to memorize everything about TSI. Okay. It might be a good idea to remember that it's not yellow if there's nothing fermented, and all yellow is both glucose and one other sugar is fermented, sucrose or lactose. All right, let's move on to the sulfide endomotility test, and I usually call that the SEMS test. This is a media that is running 3 tests at one time, and you're looking for the production of hydrogen sulfide, why it's called sulfide endomotility, looking for the production of endo, and then you're looking for the motility of the bacteria in the SEMS too. You add Kovacs reagent once again to determine whether indole is produced. So remember to add Kovacs reagent to look for the production of indole in a SEMS test. For the motility, you just look, you stab inoculate the tube, and if the bacteria only grow in the stab, it's non-motile. If the bacteria flare out from the stab or the whole tube becomes turbid, then it's motile. And then after adding the Kovacs reagent, you can look for the production of indole. If it's red at the top of the tube, then indole was produced. If it's not red at the top of the tube, then indole was not produced. Any question about any of that? So this is a negative tube, negative control, and there's no motility. Actually, I don't even see the bacteria growing there. No indole production, why it's not red, and then no hydrogen sulfide being produced, why it's not black down here. Tube 1, hydrogen sulfide is produced, that's why it's black down here. You note the top of the tube is not black. You then add Kovacs reagent to the top of the tube, and it will be reddish or pinkish if it's positive for the production of indole. And then you look to see where the bacteria are. It's hard to determine here because of the black stain, but if the bacteria, this one shows it best, if the bacteria grow outside of that stab, this one, the bacteria is only growing in the stab. This one, the bacteria, there's the stab. And we can see the bacteria growing in this region and this region here too. And so the bacteria are clearly moving away from the stab. So this one is motile. has indole formation, but no hydrogen sulfide production. All right, any question about SIMS? SIMS test? You can use McConkey's and Hectone enteric agar to look for the hydrogen sulfide production. Let me think about that. That's true for Hectone enteric agar, not for McConkey's. You can only see hydrogen sulfide being produced in Hectone enteric agar. McConkey's and Hectone enteric agar are selective. They will grow gram negatives, and they are differential. You can look at the plate and differentiate between lactose fermenters and non-lactose fermenters. If the bacteria is grown on McConkey's, meaning a gram negative, which will grow in McConkey's, and it ferments the sugar lactose, it will turn purple. If the sugar lactose cannot be fermented, then the bacteria will not be purple or pink. And this we call either white or off-white, non-lactose fermenting species. The same is the case in Hectone enteric agar. If it's non-lactose fermenting, the bacteria will be white. If it's lactose fermenting on Hectone enteric agar, the bacteria will be salmon colored, kind of an orange or reddish orange color. And if the bacteria can produce hydrogen sulfide, they will be black. Colonies will be black. All right, any questions about McConkey's agar and Hectone enteric agar? All right, on lab 9, you're not physically going to be performing any of these biochemical tests, but you are responsible for knowing how to perform and interpret them. We have a number of video clips. The first one is on carbohydrate fermentation, the carbohydrate metabolism test. We have a video on the SIMS test. We have a video on the MRVP test. And remember, this is 2 tests, the MR test and the VP test. But normally you inoculate them as 1 tube, and then split the tube into 2 tubes. Use 1 portion for the MR test, 1 portion for the VP test. We have a video on the TSI test, a video on McConkey's agar. You also have to view some of the website Microbuzz. Click on the differential test. It'll be a button on the left over here to expand the menu. You only have to look on the Microbuzz website for answering the questions on step 1 of worksheet lab 9. Let me show you that. So to fill in the worksheet, this table in the worksheet, if you don't get the answer from the video, you will have to get it from the website Microbuzz. Any question about any of that? And that link should work to the website Microbuzz. You only need to click on the tests that are needed to fill in a table in step 1 of the worksheet. They have additional links. You don't need to click on them. All right, any questions about that? And then you need to do the case studies. There are 5 case studies in this lab. So read each case study. They will tell you about the bacterial infection that somebody has. And then they will do different tests. So how the bacteria is growing on the blood agar plate, how the bacteria is growing on the McConkey's plate. And that tells you it's gram-negative that it's growing on McConkey's. It's also fermenting the sugar lactose because it's pink. There's the gram stain confirming that it's gram-negative. Let me blow that up. I think it's a bacilli. Yep, bacilli. And then here's some more tests. They did the catalase test, which isn't shown here, and they said it was catalase positive. And they did the oxidase test, which is also not shown, and it's oxidase negative. And then they did these tests. That's triple sugar iron. That's citrate. These two are the MR and the VP test, and that's the indole test. Tell you what they are. Anyways, record the results of the following biochemical tests. Oh, there it is there. So this table got broken. This is the SEMS test, the MR test, the VP test, the citrate test, and the TSI test. Record the results of that in the worksheet. I have that here. So record the results here. I remember it's indole positive. So you've got a plus right there. Okay, study 1. Record the results here, and then look up what species is causing the infection. It will be in the table Enterobacteriaceae or Pseudomonaceae, which is shown right here. Now, only use this key for the case studies in this lab. Don't use this key for your unknown project, for example. Okay, because the only species we're looking at are Pseudomonaceae and Enterobacteriaceae. All right, any questions about what you're doing? Remember to write your data in the worksheet and submit only the worksheet. If you submit the lab module, I will give you a 1-point penalty. You also need to answer the questions in section 2, complete the following. And this is asking you about the test in this lab. All right, if there's no questions, I'm going to say go ahead and get started on the lab. I will be here until 8:20 to help you with any questions. Another thing I wanted to mention, and that is over the weekend, I did grade some of the quizzes, and I also graded some of the labs, some of the lab worksheets, and let me call it up because I'm forgetting. Oh, and some of the lecture worksheets. So you should take a look at those. Point up there. For the labs, you have until this Saturday to make a correction on the labs where you had something wrong. So I went through and graded the labs and said which question, which part of the question you got wrong. And let me see which labs those are. Lab 4, I think lab 3 as well. Let me look. And I'm working on lab 5 now. I hope to have it finished by tonight. So those labs in particular have been graded. And I guess it's only lab 4 and lab 5. You can make corrections up until Saturday. Any question about any of that? All right. I also graded some of the lecture worksheets, and I think I got all of those graded. I hope to be able to get the last quiz 3A graded, not this week, but during the weekend. Okay, any questions about any of that? All right, so work on the lab.